Good afternoon, everyone. We are the Goof Tribe, and our topic is all about gene therapy. So, our group members, Kat Sandanya, Mami Ernst, Ernaline Kamada, Dayan, Princess Dayan Sagmayaw, Rayan Santos, and Rika Mitanatano. So, for the table of contents, we have introduction, types of gene therapy strategies, gene therapy approaches, methods of gene therapy, ex vivo gene therapy, in vivo gene therapy, and vector gene for gene therapy. For the introduction, gene therapy is a novel treatment method which utilizes genes or short oligonucleotide ano, sequence as therapeutic molecules instead of conventional drug compounds. This technique is widely used to treat those defective genes which contribute to disease development. It involves the introduction of one or more foreign genes into an organism to treat hereditary or acquired genetics defects. The disease is treated with minimal toxicity and by the expression of the inserted DNA by the cell machinery. In 1990, FDA for the first time approved a gene therapy experiment on ADAS-CID in the United States after the treatment of anxiety disorder. So, ayan. Ang dami na namang words, mahirap banggitin. Introduction for the part 2. Many diseases such as ADAS-CID, X-linked SCID, liver congenital amaurosis, a retinal disease, Parkinson disease, multiple myeloma, chronic and acute lymphocytic leukemia, adrenoleukodystrophy have reported of successful clinical trials. But these are not still approved by FDA. Some other disease on which gene therapy based research is going on a hemophilia, tyrosinemia, hyperbilirubinemia, cystic fibrosis, and many others cancer. After 30 years of research and clinical trials, only one product called Glybera got approval in November 2012, which was made available in market in late 2013. It has the ability to cure lipoprotein lipase deficiency or LPLD, a very rare disease. So let's proceed to the types of gene therapy. There are several approaches for correcting faulty genes, the most common being the insertion of normal gene into a specific location within the genome to replace a non-functional gene. Yan. So, first type of gene therapy. The first one is somatic gene therapy. Therapeutic genes transfer into somatic cell. Introduction of gene into bone marrow cell, blood cell, skin cell, etc. It will not be inherited later generation. At the present, all researchers directed to correct genetic defects in somatic cell. So, in germline gene therapy, therapeutic genes transferred into the germ cell. Genes introduced into egg and sperm. It is heritable and passed on to later generation. For safety, ethical, and technical reason, it's not being attempted at the present. So, ang pinagkaiba nilang dalawa, si somatic cell, ito, therapeutic genes transfer into somatic cell. Ito naman, therapeutic genes transfer into the germ cell. Then, etong somatic cell, hindi siya napapasa sa other generation. Ito naman, napapasa siya sa generation na itong germline genes therapy, napapasa siya sa ibang generation. So, ayun. Itong germline gene therapy, hindi pa siya ginagamit since para sa kapakanan ng mga gagawa ng ganitong therapy. Kailangan pa ng more lab or marami pang pag-aaral para maging successful at walang mangyari sa mga matatrial ng ganitong gene therapy. So, for the next slide, let's call Ate Ernaline or Mommy Erns for the gene therapy strategies. Thank you.